All right, good afternoon and welcome. It's 12:03, so time to begin. This is the UCCS Speaker Series. I'm Richard Kravitz, Director here at UC Center Sacramento. Welcome to our in-person audience and of course to our Zoom audience out in TV land. This is the first session of our 23-24 series on technological innovations for addressing some of California's biggest challenges. And today's talk is on mosquitoes, which are they're small insects, but that doesn't mean they're not a big challenge. They're responsible for the spread of diseases causing over 1 million deaths worldwide. And with a warming climate, previously exotic diseases like Zika, chikungunya, West Nile, malaria, and others are now real threats, even here in California. But fortunately, we have a number of existing and emerging tools to address these threats, and these tools form the basis of presentations by our two distinguished guests. First, Dr. Christopher Barker will focus on surveillance, and Dr. Kandul on technologies for safely controlling or perhaps even eliminating disease-carrying species of mosquitoes. Chris Barker is an epidemiologist and professor at the UC Davis School of Veterinary Medicine, who has expertise in vector-borne illnesses and mosquito control. He's principal investigator of the CDC-funded Pacific Southwest Regional Center of Excellence in Vector-Borne Diseases. He's published extensively in this area, and in recognition of the impact of his work was a 2019-2020 UC Davis Chancellor's Fellow. Nikolai Kandul is an affiliated research scientist at UC San Diego and co-founder of Synvec, a company devoted to mosquito control that envisions a world free from mosquito-borne disease. His research focuses on developing novel technologies to suppress or replace insect populations, such as precision-guided sterile insect technique and CRISPR-mediated gene drives to tackle insect threats to food security and the spread of vector-borne diseases worldwide and, of course, here in California. Without further ado, it's a pleasure to invite first Christopher Barker to the podium, and he'll be followed by Dr. Kandul. Chris?
Just want to say a quick thanks to Dr. Kravitz and the team here for inviting us to speak today. And I think what we do on the science and surveillance side at UC Davis and all of our UC campuses is really made more relevant by many of you in policy. And we really appreciate the opportunity to sort of intersect what we know with all of you who are interested in broader impacts and working with the legislature and regulatory authorities in our state. I just want to start off by just mentioning some of the mosquito-borne diseases in California. That we have zoonotic arboviruses, and for those of you who don't study pathogens all the time, zoonotic pathogens are pathogens that circulate in usually wildlife or other animal species and then may occasionally affect humans. And so West Nile virus is one of those viruses and our main one that we worry about in our state. We have recurrent outbreaks of West Nile virus in our state. You can see here along this axis showing the number of cases each year over California and Arizona, which I showed for comparisons, and also just to demonstrate the variability there. There's a lot of year-to-year variability in how big these problems are, how many mosquitoes we have. This was a good example of such a year where there was a lot of water in California, which is no surprise to anyone in this room, I guess. And that also brought with it a lot of mosquito habitat that resulted in actually more mosquitoes and more West Nile virus than we've had in the most recent years. The bar that really stands out there is also an outbreak that occurred in Arizona in 2021, which exemplifies what the problems can be, where you can see the orange bars across the graph are not, notably even close to as high as that one bar in 2021, and that's how these pathogens work. They can really emerge kind of out of nowhere, so to speak, or from what seemed like normal conditions and become a big problem in short periods of time. So there's a lot of monitoring that goes on to try to prevent those problems. And maybe the reason our state has more moderate variability is partly because of the reactions we have in vector control and public health to the problems when they occur. We also have St. Louis encephalitis virus, which was gone from California after 2003. There's been surveillance for it every year since, and then in 2015 it returned to our state. Very, very closely related to West Nile virus in terms of the virology, uses some of the same mosquitoes, same birds, and so in this ecological sense, they sort of compete for space in the ecosystem, and there is actually some cross immunity between the two viruses. And at least in 2003, what we thought is when West Nile arrived, that it may have actually sort of preempted St. Louis transmission to a degree that caused it to fade out. And then we've got the probability again, well, when does it come back? So it actually circulates in the southern hemisphere and in between. So it eventually recurred in California and has actually been here since. Down at the bottom right, you can see the map showing St. Louis encephalitis virus that actually made its way all the way up to Reading this year for the first time in about 50 years, which is kind of crazy when you think about the opportunities it's had in between. So there's something about the niche in the Sacramento Valley. It doesn't seem as good for St. Louis encephalitis virus as West Nile, but did make it all the way to Reading this year with all the extra water. We also have what we call 80s borne arboviruses, and the one you might know best is dengue. And I think all of you have been around long enough to probably remember Zika as well. And so these are viruses transmitted by mainly Aedes aegypti, which is a mosquito species that's really efficient at transmitting pathogens between people. And Aedes aegypti loves to live among us in our communities in cryptic habitats and backyards and places like that make it a very efficient vector of pathogens like dengue and Zika. We've also had Aedes aegypti and some other invasive species spreading in our state since 2013. And I'll show you just a bit more about that later, but we've got over 370 cities and towns that have Aedes aegypti, over 80 cities with the Asian tiger mosquito, Aedes albopictus. So both of these are problems in the sense that they can create the conditions for dengue and Zika transmission. You might have noticed in the news recently that there's been 2 recent locally acquired infections in people that didn't have travel histories to dengue endemic places. So we did have dengue transmission occurring in Pasadena and Long Beach in the LA area in September that got reported recently in October. West Nile virus transmission looks like this. So there's a number of mosquito and bird species involved, and there's some main ones. So I'm not going to go through all the details of biology for the purpose of this talk. But the way the virus gets around is actually getting in a mosquito that then bites a bird that then gets bitten by more mosquitoes. And that cycle continues. And if it gets in a human or a horse, it's a dead end for the virus. We don't develop enough of a viremia, enough of a level of virus in our blood that can infect mosquitoes. But we obviously care about human and horse populations from a disease standpoint. So that's where the disease problems start. There's some wildlife angles on this too, where birds are also impacted by West Nile and can die of it also. The virus, like most mosquito-borne pathogens, does best when it's warm, best when mosquitoes are high. It's highly seasonal. This is actually a national curve from a paper a few years ago, but the risk for West Nile virus in California and many other states is concentrated in that mid to late summer period and then drops off rapidly thereafter. We don't see this curve as it happens. So that's true in California this year, where I told you there were 299 cases of West Nile in California, but there will be more, and they're still being reported, even if they happened in September, early October, even August. We get crazy reports that happen well after crazy, I shouldn't even use the word crazy in talks like this, but we get reports that are much, much later than you would like them to be from the public health reporting standpoint. You've also, if you're in the Sacramento area, and I know some of you who are interns here may have not been in the Sacramento area in the spring, but all over our state we had good water this year. I mentioned that. The SAC-B, here's one article that they did running, basically asking is, should we be worried? All this water seems to spell trouble from a mosquito standpoint. And then we're often in that position where we know what the climate conditions are early in the season. It's not a one-to-one relationship where we just in April can tell you how bad this year is going to be. We'd love for that to be the case. But from a predictive standpoint, there's just a load of surveillance that has to happen to tell you how bad the year is as the year reveals itself. This is a look from CDPH's westnile.ca.gov website, which you can go to on your own if you like. One really nice feature of that site is CDPH compares the year so far, which is the high blue bar there, with what has been seen in previous years up to that point. Because it's nice to know how many cases occurred in total in previous years, but a better read on the year is what the year looked like at the same time in other years. And then we have that delayed reporting that I talked about. So the number of cases we've seen in California so far is 299. And then the 2022 numbers are the purple bar, and then the orange bar on the right is the 5-year average by this point in the year. So there were over 200 cases in 2022, but what you can see is that purple bar, by this time in the year in 2022, we'd only seen a little over 100 of those. So if you just project this year onto that, you might say, well, we might have only seen half of them. It could end up being 500 or 600 cases of West Nile virus by the time everything's reported in the very end. And that's the challenge for mosquito control, is it's too late to do mosquito control right now, but we're often dealing with information like this. You can see the numbers for other forms of surveillance here in birds, mosquitoes, chickens, and horses also give us some reads on that. And I would say, you know, on balance, the mosquito information and the chicken information are much better reads on how bad this year has been than the human cases at this point. Mosquitoes have a natural life cycle that consists of stages in the water. There's egg stages where the females lay their eggs, in the case of the West Nile virus vectors, on the surface of the water. Those eggs hatch, then they have these larval stages that feed in the water, so you know, diet and all of those kinds of things come into play for their success. That's also a stage at which you can target them in the water before they even emerge and bite anyone. Then we have a pupal stage that doesn't feed, and if you're familiar with like caterpillar and then the cocoon that becomes the butterfly, this is the same idea in the water. It's the larval stage becoming the pupa that doesn't feed, and it's like the cocoon, but it can move. It actually can move around quite a lot in the water, breathing through the water surface, and then eventually the female and the male mosquitoes emerge from these pupae. The females take blood, the females also rest in particular places, and then they'll eventually develop their eggs and lay eggs back in the water. The cycle repeats itself. That's a function of temperature. It gets faster in summer. We also have a spatial landscape that's somewhat challenging in some sense for mosquito-borne viruses. So we've got viruses like West Nile virus that the virus itself benefits from high mosquito numbers, of course, and we've got crops like rice. So the blue areas here from the USDA cropland data layer, and that's showing where rice is in our Central Valley here. You can see the gray areas are the urban areas. So we've got the city of Davis here, we've got Sacramento over this way, West Sac. But a lot of these populations are in pretty close proximity to irrigated agriculture. And irrigated agriculture is wonderful mosquito habitat in many cases. So from a public health standpoint, you'd like to separate the people in the agriculture. We're not a state that wants to do that, right? So we've got to get all these competing interests together. But the reality is the mosquitoes produced in this irrigated agriculture then fly into nearby communities, and that presents big challenges for mosquito control. Also in context of Dr. Kandul's talk, another thing these strategies have to think about is how far do mosquitoes fly? And if you're going to put mosquitoes out into nature for the purposes of controlling them with these novel technologies, it can be more or less challenging depending on what kind of mosquito you're targeting. Some of them don't move very far. I would say on average, those are the easier ones to do these technologies with, where you can target them in isolated areas. If they're mixing at huge scales, you've got pretty vast areas you would need to cover with some of the technologies. Also, there's loads of urban mosquito sources. So you've probably seen some of the public outreach that happens in public health and mosquito control, getting people to clean up their yards, basically. If you don't let water sit in your yard for more than a week, you don't produce mosquitoes. That's great, right? And so I'm sure everyone complies with that, right? Not exactly, right? So, I mean, I can even in my own backyard occasionally find something neglected, right? So there's a lot of these. Depends on the mosquito species as to which ones they're using. There's even underground habitats like the catch basins that deal with stormwater in our communities. There's structures under the ground that actually hold water for extended periods. And I think that's one of the reasons why even during droughts, we're happily watering our lawns in some cases. There's still water underground, still nicely producing mosquitoes that may result in human disease risk. And in fact, West Nile also has a relationship with drought in addition to the water. There's also urban mosquito movement. So as mentioned, some of these don't fly as far. If we're talking about the dengue Zika vector Aedes aegypti, that's kind of noteworthy in that it does not fly nearly as far as the West Nile vectors that transmit West Nile and St. Louis do on agricultural landscapes. Okay, here's one font issue. So I guess the font wrapped, but sorry. But what we're thinking about is this mosquito bird cycle. You've got a community of people, and the way I think about these things is if I'm walking through a park in Davis or you're walking through Sacramento, what's the rate at which you might be bitten by mosquitoes that could transmit West Nile virus to you? So that's just one way to quantify the efficiency of transmission. So this is called the entomological inoculation rate. This is obviously not a math lecture, nor is this much math here. But what we're really talking about is the number of vectors per human. So the more vectors you have per capita, the more risk you have. And that makes sense, right? If you bring one infectious individual into this room, if you have more mosquitoes that might potentially bite that person and transmit to another person, that's more risk. So the higher that is, the more efficient transmission would be. We also say, well, how often do they bite? So if you could have 1,000 vectors in this room, but if none of them are biting very frequently, that's less risk than ones that are biting very often. We also have which host do they prefer? And so from a West Nile standpoint, there's vectors that might matter because they're very efficient transmitting the virus between birds, but if they never bite a human, that's not disease risk, right? That's efficiency. West Nile might be doing great, but we're out of the cycle. But the bridge vectors, as we call them, the ones that might first bite a bird and then bite a person, are the riskiest. And unfortunately, we've got plenty of those in our Central Valley and places like LA. A lot of those are rather opportunistic, where they do bite birds and people depending on what's available to them. And then we have what fraction are capable of transmission. So if a vector becomes infected, it also has to be capable of eventually transmitting the virus in its saliva, which is a process that takes time, actually. This is what surveillance looks like in a year. So we've got this period, like I described, in the springtime, where you might be more in the realm of early warning. We haven't yet seen what's going to happen this year, but we got some rough ideas. And this was, a nice extreme year where there's so much water, it's a little easier to predict in those years. If it's a middle year where not noteworthy either way, that's much, much harder. Then we get into early detection phase, where there's a lot of surveillance in our state, lots of agencies collecting mosquitoes, quantifying those, testing those for viruses. And so at that point, you want to detect the problem as early as you can, and especially when it's above normal. Like if something's going high, you want to detect and react to that before you hit the top of this epi curve, where ultimately you're trying to avoid that human disease risk. And so for zoonoses, you get that opportunity to sort of see the process in mosquitoes and birds and ideally preempt that. And so that brings us to control, right? So the way control typically works is with action thresholds. So pretty much every agency in the state that does this kind of work, including our local Sacramento YOLO mosquito control district here, has thresholds that they look at all year long. And if those thresholds don't get exceeded, everybody's happy, they're happy, less pesticide use, all those kinds of things. And then when they do get exceeded, that's when the challenge happens, right? So they've got to react to that and try to keep that curve as low as they can to reduce the disease cases to the extent possible. And then this sort of learning cycle in the upper right's just depicting that idea that they then evaluate what they did, evaluate those action thresholds, and keep feeding that back in a continuous loop where ideally we're learning from these things and calibrating our action thresholds accordingly. Surveillance and control is what we think of as a three-legged stool in California. It's long existed this way. We've got the California Department of Public Health, who does lots of coordination efforts, reporting back to all the local agencies. They develop response plans, which they did when West Nile was moving across the country, starting in 1999, got to California in 2003. They were ahead of that and having a response plan that dictates different conditions under which you would do different sorts of actions, and escalating as you go if you start getting into human disease risk. They're also responsible for training and certification. That's tethered to the fact that certain technicians around the state are capable of applying pesticides, but to do that they have to do continuing education at regular intervals on many different topics related to both vector-borne disease, pesticide safety, all of those kinds of things. And then they also do outbreak investigation. They support the smaller communities. Okay, I'm watching the time here. And then Mosquito and Vector Control Association of California has member agencies across the state. They're responsible for the kind of boots on the ground activities that involve the surveillance, the vector control. These decisions on what to do about West Nile virus, for example, are often made at local scales. And then UC Davis and our partners are often involved in the training, research, and service. We have the Pacific Southwest Center of Excellence that Dr. Kravitz mentioned, but we also do research on the biology, the ecology, and epidemiology, and then also some of the decision support and data services, as well as the lab diagnostics. I'm going to go through this part a little bit quickly, but I mentioned the chickens, the dead birds, the mosquito pools. There's centralized testing for all of those things that's either done by California Department of Public Health or our lab at Davis. Some of that's testing for antibodies in the chickens, where you have antibody negative chickens that if they turn antibody positive, you know they got infected with West Nile in a certain window of time. Those are placed in certain parts of the state. Mosquito pools and dead birds are actually testing for the virus, so whether or not they're infected at either the time the bird died or with the time the mosquitoes were collected. We also have a data system, so this is used daily by agencies all around our state. It's known as VectorServe, and in California we call it CalServe for our variant of that. It's received a lot of support from NASA, from CDPH, NBCAC, CDC, and the state of California, so just want to acknowledge a lot of funding support for this system. We now serve additional US states as well, but we started in California in 2006. A lot of different types of surveillance and control data that are put into this system. The way it works is local agencies tend to put the data in, and the centralized labs can add the lab data. There's interactive maps and tools. So our fundamental goal for the data system is to make the data more useful. Try to give them easy ways to ask spatial and temporal questions that help guide those decisions on mosquito control. And then CDPH and CDC play reporting roles also with weekly bulletins. There's automated emails back to the agency and things like that make sure the information's in the decision maker's hands and as close to real time as possible. I mentioned the response plan. If you want to check that out, you can at westnile.ca.gov. Just click on the resources and reports. That has some of the risk levels and other activities listed in there. And then I just wanted to mention our maps. So maps.vectorserve.org, you can go on there and see how the year has progressed. And I showed this little polygon here on the map. I just drew that around that area. And what this is showing is the recent history in our area near the Sacramento and Davis areas. And you can see kind of how that stands out for this year being much higher than recent years for those environmental indicators. And again, the human cases haven't yet been fully realized. So this is what it looks like from the entomological data that were collected over that time. Now, just wanted to quickly at the end contrast with dengue and Zika virus, and I mentioned those earlier. They're pathogens that don't need birds. In fact, they prefer not to be in a bird if you're dengue virus, right? So birds are dead ends for dengue virus. We're dead ends for West Nile virus. So all you need for dengue and Zika virus is mosquitoes and people. And it's different mosquitoes, but in that case, you're often in this situation where the surveillance indicators that matter for dengue and Zika are really those human disease elements. So you're actually trying to trace cases, figure out where those people have spent significant time, and then look at maybe the mosquitoes in the area, but also potential other cases that could have occurred and been undetected. Whereas with West Nile, we sort of flip that. We're actually looking at the vectors, we're looking at the wild animals, meaning the birds, and then trying to get at the risk before it happens. So there's some pretty big differences there. I don't have time to go into all of those. I just want to mention the spread of Aedes aegypti in our state. So 2014, 2018, 2024. 2014, actually, we only knew that Aedes aegypti was in the Central Valley and the Madera-Fresno area. We knew it was in the LA Basin area. And then 2018, continuing to spread. In 2024, it's made it all the way up to the end of the Central Valley, all the way up to Redding. So that's a problem, right? That presents risk for dengue, even if dengue is not being transmitted in these places. And the other piece of this puzzle is importation risk, right? So where are the travelers coming back from dengue endemic areas where cases might occur, and how that intersects with the entomological risk from the mosquitoes that are now there. This is a look at those data. I'm just in the interest of time, I'm going to shorten this, but just because there were recent cases in the LA Basin, I picked the city of Ontario in San Bernardino County, just to show an example of a place where these gray bars indicate the collection effort over time, and the proportion of the gray bar that's filled by yellow is the fraction of the collections that detected aegypti. So what you can see is the growth in the Egypti population over that time leading into 2023. These are the latest data. But that's a growing problem, growing potential for dengue, right? So if somebody comes in with dengue, the risk you have in 2023 is much higher than it was even in recent years. So if you want to go on and explore that, you can go to that maps.vectorserve.org link and click on invasive. It tells you a little bit about the history of these different invasive mosquitoes as well, and when and where they've been detected. Dengue and Zika is something we also estimate from models. So that's that idea of the basic reproductive rate. Above 1 would sort of at least suggest that the pathogen could perpetuate itself and that one introduced case could lead to more than one additional case in a population. We've benefited a lot from COVID, where we're now all armchair epidemiologists and think you can throw around words like reproductive rates in conversation, and some people know what you're talking about anyway, but it's not an absolute threshold, but a relative indicator of when and where risk is high. In LA, you can see what the temperature gradient does as well, because it's much cooler near the ocean than it is even a few miles inland. So the risk in a place like Pasadena, I would say, is much higher for onward transmission of dengue than it is even in Santa Monica and the other areas along the coast in LA. So with that, just wanted to end with the control strategies, because that's a transition to Dr. Kandul's talk. It's a complex situation with mosquitoes and hosts. So we've got vertebrate hosts, sometimes us, sometimes birds. We've got mosquito adults, we've got mosquito immatures, loads of biology that we obviously, neither one of us can go into that in this hour. But down at the bottom, what you see is the control options available to us. And you know, in some pathogens, we have vaccines. In horses, we have vaccines for West Nile virus. I think the reality of vaccines is we'll never want to even vaccinate California's population against West Nile, probably, right? So there's trade-offs in vaccination. You don't just vaccinate for everything that's vaccine preventable. So even the development pipelines are not there with things like that. For malaria, we've got bed nets and drugs that are not really options for the viruses. So really what we're talking about is the vectors. And with that, I will turn over to Dr. Kandul, who's going to tell us all about control strategies and vectors. And I'll just leave you with the slide.
I also want to thank Dr. Kravitz and Brooke for inviting us and hosting to give a presentation. So the goal of my talk is to describe the technology we developed at UC San Diego and put that development into the context of other technologies for mosquito control. And I put two affiliations on the slide. UC San Diego, that's where all the technologies were developed, which I will be talking about today. But 2 months ago, I resigned from university and I went full-time to work for a company as a CEO, a company called Synvact. And the goal of this company is to bring these technologies into the field. And that's my kind of conflict of interest disclosure. Mosquito is by far the most deadly animal. You kind of already saw the picture. Some statisticians estimate that 50% of people since the dawn of humanity died due to mosquitoes. One mosquito in particular, Aedes aegypti, yellow fever mosquito, is the most invasive mosquito. Its Latin name Aegypta, it's from Egypt, but it's spread everywhere, kind of where it can live. And it poses a threat of transmission for dengue fever, yellow fever, Zika, chikungunya, West Nile, and many other diseases. And that basically threat now for 4 billion people around the world, including people in California. And global mosquito burden will only expect it to increase because of a global warming, globalization, and insecticides resistance. And these are just kind of 3 newses I grabbed from the internet. So there are 2 local transmission of dengue in California, plus this last summer, Florida had a record number of malaria cases. There was 8 cases, again, local transmission of malaria in Florida. And then Paris was fumigating mosquitoes inside the city. And Paris will have Olympic Games next summer, so they really have to kind of get on to mosquito control. There are many approaches to control mosquitoes and disease spread. And this slide describes basically 5 different categories, but they all have their own limitations and shortcomings. Biological methods of controlling mosquitoes, probably the safest and the most promising approaches, where you essentially kill a mosquito. And if you kill a mosquito, you don't have a vector who transmitted disease, and you stop the disease transmission. But historically, biological methods for mosquito control were difficult to scale and deploy. The sterile insect technique, or SIT, has been the most effective and sustainable biological method control of all insects. And in SIT, essentially radiation or ionizing radiation is used to irradiate insects, to sterilize them. And then these sterile insects are released into an environment where sterile males will search and mate with wild-type females. And because males are sterile, and many insect females, they mate only once in a lifetime. So if a sterile male mated with female, that female will stay sterile as well. And that's basically how this approach works. And it was used very successfully in 1950s in US to control New World screwworm fly. And this program was very successful, and this fly was completely eradicated from southern states of US, then in Mexico, and then in Central America. And one note that for SAT program of New World screwworm flies, both sexes, males and females, were released. And they are still released, actually, on the bridge between Colombia and Panama, just to prevent the introduction of these species back into Northern America from South America. Mosquitoes, only female mosquitoes bite and transmit disease. Male mosquitoes, they look differently from mosquitoes. They don't bite at all. Therefore, female removal, or I call it sex sorting, is required for mosquito SIT programs, especially if you're releasing these huge number of mosquitoes over a residential area. This slide also mentions another method called Wolbachia-induced cytoplasmic incompatibility, and sometimes the method is called Wolbachia-induced incompatible insect technique. Essentially, Wolbachia is a bacterial symbiont, and if you have males which carry Wolbachia, but wild-type females don't have this Wolbachia, then they mate together, cytoplasmic incompatibility happens, and the legs do not mature. So there's this technology which is called WIIT, Wolbachia incompatible insect technique. Works very similar to SIT. And for both of these technologies, you have to sex sort mosquitoes. You have to remove females before you're releasing male mosquitoes. And both of these technologies are quite old. But there are three big challenges which prevent the application of these technologies, SIT and Wolbachia IIT, for mosquito control. And the first one is effective sex sorting, or removing female mosquitoes. Traditionally, it was done by hand under microscope, and obviously you don't have a scale, you don't have production scale to release so many males. And then there is also economical efficiency. Male mosquitoes are quite fragile. You cannot ship them. You basically have to produce them locally. So that means you have to invest capital money into building facilities close to the places where you plan to release mosquito males. And then you need to have continuous production of these males because you cannot keep them, you cannot store them. And then in addition to that, 50% of your production capacity will be essentially sacrificed because it will be females. Then another problem is the suppression efficacy, because radiation or Wolbachia are used as a sterilizing agent of mosquitoes, but they also change fitness of mosquitoes, and mosquito males which you release in the environment, they are not very fit or competitive, and they don't have longevity long enough. But they have to compete with wild-type male mosquitoes for mating with females. And then what it means, you have to deploy really massive numbers of mosquitoes, 10 times more at the minimum for these programs to work and for you to see some suppression. So this slide is very complicated, but it basically outlines all the current technologies available for mosquito control. And I divide it into two groups: confined technologies or non-gene drive in green, and then spreading gene drive technologies. And for confined gene technologies, what it means on the graph, you can see there is triangles. You have to deploy your solution multiple times to see the suppression of population. And SIT and Valbachia incompatible insect technique belong to these suppression methods, as well as Oxitec genetically modified mosquitoes or GM mosquitoes. They also belong to this category. But then there is a completely different methods which are essentially self-spreading or self-propagating. And then you can divide them into two groups. One of them will be modification of a wild type population, where you release genetically modified mosquitoes, and those mosquitoes are resistant to certain diseases, and they actively spreading. They're spreading their gene in the population. And that would be modification. And then there is a suppression, where you're spreading certain genes you kind of develop in the lab, but those genes cause female sterility, for example, but you can still spread them through males. And that would be gene drive, which will cause suppression of the population. And ideally, gene drive technologies is good because you, in theory, should be deploying them only once, because they will basically self-catalytic, they'll do the job. The problem with it is basically, you don't know where your gene drive mosquito will end. In theory, it will spread in places where you didn't intend it to spread. But more importantly, scientifically, there is not a single gene drive constructed which actually can effectively spread itself in a population, even in the lab. Because eventually it will meet the resistance alleles which will stall its spread. So it's not really effective. But in addition, gene drive technologies, they face severe regulatory hurdles because it's something new and it's made to actually be super GMO. So that's why a lot of researchers, they turn to the older technologies as sterile insect technique and Valbachia compatible insect technique, and they try to engineer solutions around. And these pictures show like two ways how you can sex-sort mosquitoes which belong to two genera, Culex and Aedes. Those genera of mosquitoes, they have pupae of different sizes. Females are much bigger than males. So that means, as a first step, you can use literally sieve and to remove majority of females as a pupil at the pupal stage. And then you'll have maybe 2 or 5% of smaller sized females still remaining in the culture. And for that, essentially, that's where engineering came into the effect. And Verily, which is life science Google Corporation, they engineered the whole kind of a salute of robotic rearing of mosquitoes where the whole culture is synchronized. And then mosquitoes have to emerge and walk essentially in a procession, one at a time, as shown in the picture. And then optical system, which is trained by artificial intelligence, will see if it's male or female and will remove female. But obviously, it's a kind of very expensive and solution and not available for everybody else because it demands investment of a big capital, and it's also proprietary technology. So we need some other better solutions how we can sex-sort them. And we hope that these solutions can be also ported in other genera of mosquitoes where you don't have difference in pupil sizes, because this approach will not work for Anopheles mosquitoes. So that's where basically we came into the game as geneticists. We engineer a genetic way how we can sex-sort mosquitoes. And this system works very well, and now we have it working in 2 different genera, in Aedes as well as in Anopheles. We call it separator, and this picture shows separator genetic cassette. It expresses 2 fluorescent proteins. One fluorescent protein, DS-RAD, expresses in males and females. Now the other one, GFP, expresses correctly only in the males. You see expression of GFP only in the male, and pictures at the bottom show female and male lavis. We inserted the intron and site coding sequence of GFP, and that intron splices correctly only in the males. And this system allows us to select males using GFP very early in development. And this slide just shows you that GFP and DS-RAD, they are fluorescent proteins, but they are non-toxic, inert, and are very safe markers. They are one of the oldest fluorescent proteins available for research, and they have record of maybe 25 years where the in vivo models of mice, rats being created where these proteins expressed in the most sensitive tissue, like dopaminergic neurons, without any obvious effect on the physiology of the animal. In addition, DS-RAD also was reviewed recently by FDA and raised no concern as a marker for GM rice, which is basically GM corn, which is destined for human consumption. So they are very safe. The other cool thing about separator, because larvae are aquatic stage for mosquito, we can use large particle flow cytometry to sex sort larvae based on presence of GFP or absence of GFP. And we can do it at the rate of 10 to 20 larvae per second. And then another benefit of separator is that if we're removing females very early in development at the first level stages, so we're freeing all the resources to raising more male larvae under the same capacity. And all these facilities, which are built for rearing mosquitoes for SAT application, they usually built for a certain scale of production. So here with separator use, we can essentially double that production. And this is how separator compares to available methods, which is used by SIT or Slovakia IIT. One of them is based on the Virali technology, the other one based on Senesio robotics, but it's the same deal. You have to kind of use some optical recognitions to remove females as adult stage, where we can do it very, very early in development, and we can even release Slovakia carrying mosquitoes as a larvae. So right now we build separator in two species of mosquitoes, Aedes aegypti and Anopheles stephensi. Anopheles stephensi, it's an invasive urban mosquito which spread malaria. It's originally from Asia, but now it's spreading in Africa. So it's a major vector of malaria. And just in summary, so separator is a very cost-effective and high-throughput, high-precision sex sorting system, which let us select males very early in stage and conserve resources for rearing more males. It built on robust science. It's in a way unbreakable system where we're using positive selection to select males. And the other interesting thing that if we use separator only for SAT or WIIT, it's not considered to be biopesticide because it's not active ingredient, it's not causing any sterility. We hope it's going to be considered as a modification to production process. So I show you how we now solve the sex sorting challenge for these older technologies, but we still have economical efficiency and suppression efficiency. And to solve those issues, we use CRISPR technology. We developed the method we call precision-guided SAT. In CRISPR technology, Cas9, which is protein and works as a molecular scissors to go and cut specific DNA or RNA fragments, and then it uses a molecule which is called G or guide RNA. That molecule guides that Cas9 to specific locations. But Cas9 by itself without guide RNA is not active. So what we've done, we separated Cas9 and guide RNA, and we built two different strains of mosquitoes, one of them expressing Cas9 and guide RNA, when we can maintain them as homozygous lines. And guide RNA line have expressing multiple guide RNA targeting two genes, one of them essential for female viability, another one essential for male sterility. So now then we do genetic cross between these two lines, these components will find each other. And in this progeny, we call it F1 progeny, CRISPR system will become active and we have autonomous female removal and male sterilization. And it works incredibly effective. And this diagram also shows that for Aedes aegypti, which mosquito, like the most invasive, because you can desiccate the eggs of these mosquitoes, and eggs will hatch sometimes 1 year later, and there are some records in literature they can hatch 5 years later. But what it means for us, we can make a cross, produce a lot of eggs, dry them up, and ship them globally, because mosquito eggs are tiny. We have a global logistics with this approach. The other very important thing to mention that because we're knocking out two genes extremely specifically, the males which are produced by this technology, they are completely sterile, but they are very fit, they are competitive, and they have the same longevity as a wild type mosquitoes. So PGSAT or precision guided SAT is a safe, economical, and scalable technology. It's safe because we understand molecular mechanism, how it works. And we know how male sterility and female removal is done. And we also know that our technology consistently leads to the same outcome. Male are completely sterile, at the same time it's very fit and competitive. And then we're also producing eggs. With eggs, we have much higher production, at least 100 times, if you compare it to producing adult mosquitoes. And we can also deploy eggs in the environment, which is also much more efficient and effective. And finally, this technology works in different insect and different mosquito species. We can port it very easily into different species. And in fact, in the lab, we already developed PGSIT for Aedes aegypti and Anopheles gambia, and we're preparing the strains for 5 other major mosquito vectors. So, and finally, we also, right now, we're putting 2 systems together. We're putting separator with PGSIT. So now we have Cas9, which will have separator genetic cassette where we can sex sort, and guide array will have a separator, which will let us to do very efficient sex sorting of these strains, and then have a capacity to do very massive cross to produce a lot of eggs. And then female removal and their fertilization still happens when Cas9 and guide array will meet each other in a genetic cross. And mated females produce many batches of eggs, and these eggs can be stored and shipped for Aedes aegypti. And it's still precision-guided technology because knockout is very highly specific. It's only 2 genes side effect. And so next generation SIT overcomes every technical and economical barrier of alternative confined approaches for mosquito control. And kind of here I put them on a slide. The end result is completely sterile mosquitoes, and only males, which don't bite. We have a huge scale of production, more than 100x. We have a global logistics where we can deploy these eggs. And the sex sorting system now genetically integrated in comparison to some older technologies where you have to deploy adult male. The production cost is very high because the scale of production is 0.5x. So now about regulation. Genetically modified mosquitoes are classified as a biopesticide, and they are under jurisdiction of Environmental Protection Agency at the federal level and California Department of Pesticide Regulation at the California level. And essentially now what we're doing, we secured funding from Bill and Melinda Gates Foundation, and we are preparing both of these technology to go for regulatory framework so we can actually use it. But regulation is a very slow process, and the last slide is I put here just because there might be some people who will go into regulation and legislation. I would say that EPA and CDPR are very undecisive about confined genetically modified solutions for mosquito control, and they have been since 2018. And we are starting to lose the fight for mosquito-borne diseases. They completely understand it. They do want to go away from chemical pesticides. And in fact, 1 month ago, they had a workshop where I also presented, described our technologies. But it's a very slow process. And this is the last slide. I just want to acknowledge many people from Walmart Biolab at UC San Diego and many funders who helped to fund us and conduct research. And thank you so much for your attention.
Today, we'll go ahead and let Director stop our recording there.